Alright, so I'm going to take you through a process called excitation contraction coupling, and that's how Bob here is going to contract his muscle. So it's all going to start with the brain. It's going to be an impulse from the brain, and it's going to be from this motor neuron. It's going to create an action potential that goes down to the synaptic knob or synaptic cleft. And these little synaptic vesicles hold ACH inside of them, but once this action potential reaches the synaptic knob, it's going to allow calcium to flow in, and it'll cause these synaptic vesicles to exocytose the content inside, which is ACH. That ACH will then bind to these ACH receptors, which are ligand-gated channels. So once it binds to the ligand-gated channel, it'll allow sodium to flow in, potassium to flow out, and it'll cause an end-plate potential. And once it reaches the threshold, it'll cause depolarization in the cell, or the sarcoplasm, which is basically the cytoplasm of this muscle fiber, and it'll cause the action potential to go down the sarcolemma, which is the excitation part of the excitation contraction coupling. It'll go down the T-tubule, and a T-tubule with two cristae of the sarcoplasmic reticulum form a triad. So once it goes down, it'll activate this rhymidine receptor, which will change shape and allow calcium to flow into the sarcoplasm. So once that happens, that calcium is going to want to bind to troponin. But first, let me lay down a foundation. So first, this is going to be myosin, and this is inside the sarcomere, which is the smallest part of the muscle fiber. So this is myosin, which is the thick filament, actin, which is the thin filament, which is this red right here, and this is the M line. It's going to be a reference throughout the video. So the sarcoplasmic will exocytose the calcium. The calcium will bind to troponin, which is on actin. It'll change shape, allowing the tropomyosin to reveal and rotate. So it'll reveal the active sites on the actin. And once the myosin binds to the actin, it creates a cross-bridge formation. But throughout that cross-bridge formation, you're going to have the contraction cycle of the excitation-contraction process. So first, you're going to have, in each myosin head, you have ADP and a PI, which is an inorganic phosphate. So that myosin head is now active, so it's going to bind to the active site on actin. Once that happens, the inorganic phosphate will be taken off, and so will the ADP. It'll move the actin closer to the M line, and that is known as the power stroke in the contraction cycle. Then you have, so it's attached to the actin right now, and you want it to detach, which you need ATP for. Once the ADP is present, it'll detach, but once it wants to go through the cycle again to move these actin closer to create the contraction, you need to go through ATP hydrolysis, which will break down ATP into ADP and PI, which is the inorganic phosphate, make the myosin head active, and then it'll go through the process again, therefore creating these actin to move closer to the M line, and the sarcomere will contract, therefore causing the muscle to contract overall. So that's how you get a contracted muscle. That's excitation contraction coupling.